Thank you very much, Sigal. It's good to be back. And thank you, everyone, for coming on such a cold, cold day. So what I'm going to be talking to you about today is how animal behavior is influenced by the activities that humans do in the environment. So here is a satellite image of Earth as it appears at night. And from the light pollution distributions, you can see patterns of human habitation worldwide. And I'm showing you this image just to highlight that even though when we think about human impacts on the environment, we may think of a few specific examples, we may think of a few local effects, but it's important to remember that these effects overall are a global phenomenon. And they've really come to the forefront mostly in the past 100 to 200 years, which is a blink of an eye in terms of the age of the Earth itself. So today I'm first going to just talk a little bit about how humans have been acting to change Earth's environments. I'm then going to introduce some examples of how animals have been responding to these effects in terms of changes in their behavior. I then want to bring in a little bit of consideration to the effect that time has on the animals' responses to these changes in Earth's environments. And then I want to get into a case study based on my own PhD research here at McMaster University on the fish called the round goby in Hamilton Harbor. And then I want to kind of conclude with a prospectus to how the field of behavior and toxicology is moving forward into the future. So some of the human activities that go on actually act to confuse animals in the environment. And a great example of this is light pollution. Excess light is a particular problem at nighttime in urban areas. And this excess light can serve as a false cue. It can confuse animals by interrupting migration patterns, by giving them a false navigation cue. And it can also serve to disrupt timing of events in life cycles like reproduction. Habitat loss is another way that humans can act to affect Earth's environments. And in this case, here's habitat loss mediated through climate change. And the iconic example of this is a polar bear who is facing the disappearing Arctic ice habitats. Now, habitat loss can, of course, be directly lethal for animals, but it can also cause more subtle changes in their behavior, affecting migration patterns, natural distributions, predator-prey interactions, and the kinds of food that are available to them. And in the case of polar bears who are facing the effects of climate change and these disappearing ice flows in the Arctic, they're actually undergoing a change in diet from a sea-based diet, heavy on emphasis on predation of seals, to a more land-based or terrestrial diet, caribou. Another way that humans' activities can affect animal behavior is through the alteration of community structures. For example, the elimination of species or the introduction of species that are called exotic or invasive species. And the example that I have illustrated here are Asian carp. These are 4 species of Asian carp that have become established and invasive in the Mississippi River Basin. And you may have seen them appear in the news lately because people are extremely concerned about them continuing to move up the Mississippi River Basin and threaten the Great Lakes. And invasive species can cause a lot of problems for native animals in the environments that they colonize. They can drive them out of preferred habitat. They can interrupt existing predator-prey dynamics. They can outcompete them for food resources. In many ways, invasive species pose many problems for natives. But by and large, when we think about impacts of human activities on the environment, what many people think first about is chemical pollution. And I'm going to go through a few examples of the types of chemical pollution that are present in many different kinds of environments. And the first example is that of intentional chemical discharge. And we see this in circumstances where pesticides, for example, are being discharged over an agricultural field, or wastewater effluents that are being deliberately reintroduced into the waterways. But a lot of chemical discharge examples come from situations that are actually accidental. And sometimes these accidental examples are quite catastrophic in nature. We can think about oil spills in the ocean. For example, the Exxon Valdez oil spill that was so famous back in the 1980s, or the much more recent Gulf oil spill following the Deepwater Horizon explosion in April of 2010, which is illustrated here. And in this particular instance, the oil spill was actually made worse by the intentional application of chemical dispersants called Corexit, which actually increases the toxicity of the oil to marine organisms. But there are other kinds of chemical pollution that are perhaps less obvious, or things that we don't think about very often. Pharmaceuticals and personal care products are now found in increasing concentrations in urban wastewater. And they get into our wastewater because when a person takes a medication, we're excreting not only that medication, but also its biologically active metabolites. And these categories of contaminants have only really been recently recognized in the past 1 to 2 decades. So they're a kind of emerging class of contaminants of concern. So overall, there are a huge variety of ways that humans can act to affect Earth's environments, and I've highlighted just a few here. But for the rest of this talk, I'm actually going to focus primarily on chemical contamination. And I'm going to do so in the context of aquatic environments. This is because this is the area in which most toxicology research is done, and it's also the area that I do my own research. So next I'm going to talk about how animals are responding in terms of behavioral changes to all these modifications that are going on in the ecosystems. Think of a living organism as a complex machine with many interacting, interdependent parts. With an exposure to a pollutant, a toxicant, or any kind of chemical substance, you can think of that acting as a wrench in the machine, which can have widespread and sometimes hard to predict consequences. And sometimes those consequences are going to be lethal for the animals. But many times, an animal that's been exposed to a pollutant is not going to die. It's going to instead exhibit many sublethal effects. And the behavioral changes that I'm going to be talking about are examples of these kinds of sublethal effects. So behavior is an attribute of an individual organism. It will act to integrate any effect that is happening at the physiological level, effects on DNA, effects on cells, and effects on organs and tissues. And behavior is also the interface through which an individual animal interacts with its environment, predators and prey, other members of its own species, and natural resources such as habitat. So an animal's behavior is what defines its ecological role. Exposed individuals who exhibit sublethal effects may actually become compromised in a natural setting compared to unexposed individuals. For example, they may have a harder time growing normally, they may get sick more often, they may be more vulnerable to predators, and the net effect would be a reduced likelihood of reproducing successfully. And when this happens, from the perspective of ecology and evolution, this animal is already functionally dead. And this term has been referred to by toxicologists as ecological death. And I'm going to go through some examples of how ecological death can be mediated by chemical exposure. Some chemical contaminants can actually interfere directly with animal sensory systems. For example, dissolved heavy metals in the water like cadmium, or pesticides in the water like diazinon, can actually knock out the ability of fish to smell. And you may think that fish may not have much to smell in the water, but there are many species of fish who need to rely on their sense of smell to let them know when it's dangerous. Certain species are able to detect a chemical cue, think of it like a distress signal, when other members of its own species are being eaten. And when they smell that cue, they know it's time to hide. If their ability to smell has been knocked out, they no longer can sense that distress cue, and they'll no longer appropriately respond. And species like trout and salmon also need to use their sense of smell to guide them appropriately on their migrations to spawning grounds. So if that sensory system has been disrupted, they're no longer going to be able to be as successful. And some of that research was actually done right here at McMaster University. Other kinds of animals need to rely on learning ability to function normally in their environment. For example, honeybees need to rely on learning to properly associate sensory cues with food resources. Honeybees that have been exposed to sublethal levels of many kinds of pesticides show a reduced ability to form associations between a sensory cue and a food reward in laboratory tests. And they also forget these associations much more quickly. In a natural environment, honeybees that are no longer able to successfully pair a cue with a food reward in the field are no longer going to be able to contribute as well to the success of the hive, and the hive's success as a whole will be compromised. Some other kinds of behaviors that may be disrupted with exposure to chemical pollutants are those associated with reproduction. For example, parental care. Glaucous gulls that have been exposed to high levels of PCBs spend less time with their nestlings feeding them than gulls that have had a lower exposure. And for gull nestlings, having a reduced feeding from the parents and a reduced attentiveness at the nest will lead to overall lower growth rates for those chicks and a lower likelihood that they themselves will survive to reproduce. But sometimes the situation gets a lot more complicated. For example, there are certain scenarios where we can envision that the negative effects of contaminant exposure will actually act to cancel each other out. Red-spotted newts become really bad at catching their prey, frog tadpoles, when they've been exposed to chemicals like the pesticide carbaryl. But tadpoles at the same time also become really bad at escaping from predators when they've been exposed to the same pesticide. So when you look at the overall predation rate in a predator-prey system where the predator and prey have both been exposed and compare that to the rates of unexposed populations, there's no net change. The negative effects of contaminant exposure in this system actually act to cancel each other out. And sometimes animals are even able to detect and respond to the presence of the pollutants themselves. Some fish are able to sense the presence of certain contaminants like wastewater effluents or dissolved heavy metals, and they'll avoid those areas. And this is a protective effect for them. They will be avoiding areas that present to them an unpleasant or a noxious stimulus. And sometimes fish will actually be attracted to pollutant plumes in nature, and this will act to increase their likelihood of suffering negative consequences of exposure. So now I've just briefly highlighted some of the many ways that contaminants act to affect animal behavior. And now I want to just bring in the concept of time in how we consider how these effects manifest in both the short term and the long term. In the laboratory, the most common paradigm is the use of short-term tests, and scientists call these studies acute studies. The traditional length for short-term studies in toxicology is 96 hours, and there's nothing magical about 96 hours. It just happens to fit very conveniently in a Monday to Friday work week. Most lab tests also usually use only one type of contaminant at a time. When we see behavioral changes in the laboratory, the most frequently reported change is that of an alteration in general activity level. Sometimes animals actually become agitated. They become hyperactive. And in other cases, they may have a reduced level of activity. They become sluggish and slow. But there's any number of other responses that have been reported in the literature across a wide variety of species in these types of short-term tests. Alterations to feeding, alterations to learning ability, social interactions like aggression, reproductive behaviors like courtship and parental care, effects on behaviors that will affect their vulnerability to predators like reaction times and alarm responses. And the overall pattern is that these behavioral changes are generally interpreted as something maladaptive. They're going to impair the ability of these animals to survive in the field. Sometimes these short-term studies are accurately representing what's actually happening in the environment. For example, think about a pesticide that's been sprayed on an agricultural crop. Every time it rains, pesticide is going to be washed from the fields into local waterways, and the animals living in those areas are going to be subject to pulses of natural exposure. And these pulses are acute or very short-term in nature, but they can be quite disruptive, especially if they're happening during key life cycle events, such as a spawning event. But when we actually think about what we want to know, what's happening in the field, we're usually interested in the long-term consequences, either because we're worried about a source of contamination that is particularly long-lasting or persistent, or because we really want to know what's happening to animals over the course of their entire lifespan. And one way to think about these differences is to go back to a very old idea in science with a very long history, and it's called the general adaptation syndrome. Think of an animal's ability to resist stress to be at a certain level under normal conditions. When you subject that animal to a stressor, and it could be any type of stressor, but let's go with chemical pollution here, the body will go into what's called the alarm phase of the response. And the general result of that alarm phase response is that the body becomes less able to resist stressors. It may become more likely to be sick, for example, because the immune system has been compromised. But as time progresses, the body develops adjustments to compensate for the increased costs that the stressor is exerting on the body. And this is termed the resistance phase. Over time, individuals may actually recover from this stressor, particularly if it's removed. But if not, even over the longer term, it's possible to see entire populations becoming adapted to cope better with this stressor. But if the stressor continues to persist past a certain point, the body's ability to devote energy to coping with the stressor will actually become exhausted. Because coping with stressors is costly, it takes energy away from normal activities like growth and reproduction. And if it reaches this point and the body's energy levels become exhausted, the end result could be death. So no matter what the ultimate endpoint is of this entire stress response that we're thinking about here, from the perspective of scientists interested in behavior, it's important to remember that the types of behavioral symptoms of exposure are going to differ at different points of the exposure cycle. In some cases, the same behavioral symptom may persist throughout. But in other cases, the pattern may invert or even disappear entirely. And a good example of this comes from a study using young rainbow trout that were exposed to copper through their diet. And what I've got graphed here is the length of time that the experiment lasted against their general activity levels measured as their swimming speed. So in the initial phases of the experiment, what we see are that copper-fed individuals became hyperactive. They became agitated. They started swimming faster than the control fish. But as time went on and the exposure persisted, what we see is that the change in activity levels actually inverts with time. Fish that are exposed to copper become sluggish and lazy. And the scientists who did this study discovered that this was a protective effect for the fish. They were saving energy by not moving very much, so they could devote more energy resources to coping with the toxicity of the copper. And this allowed them to actually continue to grow normally. So how do we cope with this problem? One possible solution to this problem is to simply look at running longer laboratory studies. And some studies can even cover the entire life cycle of the animal in question. And these studies are certainly valuable, but it can be very hard to extrapolate from lab studies to complex field situations. Lab studies are run under highly controlled conditions. They're perfect, they're ideal for that test organism. And these studies tend to use the same model species over and over and over again, which may be very difficult to extrapolate to the field where conditions are a lot more messy and you're going to have a lot of unusual species living in those habitats. So another way to get at this question is to actually go into the field and look at what's happening to the animals that are actually living in a site of concern. And these studies are certainly ecologically relevant. They're using the right species that happen to be living in that area, and they're looking at the consequences of long-term exposure to sometimes very complex mixtures of contaminants because you rarely find only one contaminant problem at a time in a particular site. But every site is different, and it's a little bit harder to show cause and effect links in the field because every site is different. There are so many more variables to control. And the ideal situation is you're going to have information from both the lab and the field to combine to show that toxic effects manifesting as changes in behavior are both possible and actually happening in the wild. And now I want to just get into this case study that's part of my PhD research here at McMaster University, looking at one species of fish called the round goby in a very local area, Hamilton Harbor. So I have here a satellite image of the Great Lakes showing the border between Canada and the United States. And I have Hamilton illustrated here in yellow. So the North American Great Lakes are the largest series of freshwater lakes in the world. 21% of Earth's freshwater resources are located in these lakes, and 84% of the freshwater in North America alone is located here. They have been intensely settled for centuries and have also served as a major highway for transportation and trade. And one of the kind of unfortunate consequences of this emphasis on the shipping industry in the Great Lakes has come through the use of ballast water. So when a cargo ship needs to balance its weight, it will take up water from whatever harbor it happens to be in at the time. This is called ballast water. And then when it's ready to take on cargo, it will discharge that water into whatever harbor, again, it happens to be in at that time, even if it's traveled entire oceans in between emptying and taking up cargo. And global trade routes mean that for decades, water from all over the world was being brought to the Great Lakes and dumped. And this means that any organisms that happened to be in the ballast water at the time were also being transported into the Great Lakes. And this is the reason why we have zebra mussels and quagga mussels, as well as some other species which you may not be aware of, such as spiny and fishhook water fleas in the Great Lakes. Oh, and by the way, this ballast water practice it's no longer allowed. But one of the other species that has been bequeathed to us by this ballast water transfer is this fish, the round goby. It's actually native to Eastern Europe, and it was first discovered here in 1990. This is a relatively small animal. It's about 5 to 10 centimeters long. It's a bottom-dwelling fish, so it likes to live at the bottom of lakes and streams. It does not swim up in the water column gracefully like you might think normal fish would, but it moves around by hopping. And the round goby is a natural predator of zebra mussels. Both stem from the same area of Eastern Europe. And this is one of the main reasons why round goby have become so successful and so well established in the Great Lakes, is their ability to eat the zebra mussels. But of course, like many fish, they'll eat anything that they can fit into their mouths. And scientists are of course really concerned about the effects that round goby has as an invasive species in the Great Lakes. But for the rest of this talk, I'm going to actually focus on how this fish responds to pollution. The round goby, because it's a bottom-dwelling fish, is going to be exposed to any contaminants that are occupied in the sediment, or that are sediment-bound. And because it eats organisms that live in the sediment, it's also potentially exposed to contaminants through its diet. And this means that the round goby can actually be used as a living tool. It's called a sentinel species. And a sentinel species, just like this soldier here keeping watch, it's a tool that you can use to keep watch over the environment. You can look to the condition of this animal in its natural habitat and see how the conditions of the environment are doing in that particular location. Think about the concept of a canary in a coal mine. Back in the 19th and 20th centuries, coal miners would bring a canary with them into the mines while they worked because the canary was their living carbon monoxide detector. If the canary started showing signs of distress, the miners would be alerted to the fact that there was probably carbon monoxide around and that they needed to leave. So a sentinel species is kind of like a natural canary. We can use them to understand what's happening in the environment. So since their first introduction in 1990, in Lake St. First discovery in the Great Lakes in 1990, the round goby has actually spread throughout all 5 Great Lakes, and they're incredibly abundant. There are at least, I think, 3 billion estimated in Lake Erie alone. And these Great Lakes are also the location of many highly impacted, highly polluted sites. And back in the 1980s, when awareness of pollution was really coming to the forefront, the Canadian and US governments formed what's called the International Joint Commission, the IJC. And they sat down together and identified 43 areas in the Great Lakes that were particularly awful. They're called areas of concern, and they're areas that the governments wanted to target to remediate them. Since the 1980s, a few of these locations have been successfully delisted. They've been successfully cleaned up, remediated. But one of the areas that is still remaining to be successfully cleaned up is the AOC, or the area of concern where we live now, and that's Hamilton Harbor. So this is a satellite image of Hamilton Harbor as it appears in the present day. And McMaster University is located here over to the lower left. If we graph onto this the shape of the harbor as it existed 200 years ago, we can see that there has been substantial modifications to the natural shoreline of the harbor, mostly focusing in the southeastern portion of the harbor, where Hamilton has its renowned steel and iron industries. And there are also locations along the furthest eastern shore there, where there are confined disposal facilities for sediments that have been dredged from the harbor. Since the designation of Hamilton Harbor as an area of concern, there has been about 3 decades of research understanding what contaminants are a problem in Hamilton and where they're located. And a lot of the research that has been going on has been looking in the sediments in the bottom of Hamilton Harbor to know what's bound there. And I've graphed here some fairly recent data on Hamilton Harbor sediments that were tested for a range of contaminants. And this is just a few of the many contaminants that are studied. A few metals, cadmium, copper, lead, mercury, and nickel, and some organic contaminants, PCBs and what are called PAHs, and I'll introduce those in a moment. So the redder the dot, the more of those contaminants exceed federal or provincial water quality guidelines. So the redder the dot, the hotter the spot, the more toxic that location is. And conversely, the whiter the dot, the less toxic it is, and the cleaner that particular location is. So we can see that contamination in Hamilton Harbor tends to focus in 2 general areas. And I've highlighted them here in yellow, and they're around the industrial sector. The furthest right is Windermere Arm. This is a high-use shipping channel that has a lot of highly contaminated industrial slips. And it's also the route through which Hamilton Harbor's wastewater treatment plant effluents flow back into the harbor. The site in the middle is actually a site known as Randall Reef. And you may have heard about Randall Reef in the news. Randall Reef is a massive pile of coal tar that was left over from the steel industries when dumping in the harbor was allowed up to 1970. It is now the single most contaminated aquatic location in Canada. It used to be in 2nd place, but Sydney, Nova Scotia has now cleaned up their tar ponds, so we're number 1. Coal tar is rich in these incredibly toxic, carcinogenic, endocrine-disrupting, dangerous compounds called polycyclic aromatic hydrocarbons, or PAHs. This site has been deemed far too dangerous to dredge. We can't just dig it up and cart it somewhere because there's nowhere safe to put it. So the plan for many, many years has been to simply cap the site, build a permanent containment facility over it. And if you've been following the news, maybe you might remember back in the fall, the funding for this project was successfully obtained, this multi-million-dollar facility. As the funding has been obtained and construction will be begun soon. I think the target completion date is 2020. So the ABLE Laboratory Group at McMaster University has been studying the round goby population in Hamilton Harbor since 2002. And when I first started my PhD in 2006, one of the first questions that we asked were how were round gobies responding to these high levels of contamination in Hamilton Harbor? So what we did is we went to the harbor and we collected fish in 2 different areas along the North Shore in gray. Those are our low contamination sites. And we also went to the 2 highly contaminated hotspots in Hamilton Harbor, Windermere Arm and Randall Reef, and collected fish there as well. And when we collect round goby, we don't have to actually go into the water to collect them. What we do is we set traps for them. These are called minnow traps, and we can fill them with frozen corn, of all things, because these fish really seem to like corn. And we put the traps in the water, leave them overnight, come back the next day, and voila, we have fish. And we find that this is a really effective, easy way to get large numbers of round goby. So we were interested in understanding signatures of contaminant exposure. And these signatures of contaminant exposure are called by scientists biomarkers. It's a red flag that says, I've been exposed. And when we looked at our contaminated site fish, we first found that they're actually really abundant. There isn't much difference in abundance between areas of high contamination and areas of low contamination in terms of numbers. So these fish are clearly able to handle living in those areas. But fish from the contaminated sites do show some changes. They're physically smaller. We believe that this is associated with them also being younger. There's probably higher adult mortality at these locations. When we look at the fish's tissues, we see higher concentrations of metals like copper and cadmium accumulating in their liver. We've seen higher concentrations of PCBs in their bodies. And we do also see increased production of detoxification enzymes in their liver. These are known to be stimulated by exposure to those contaminants, those PAHs that I talked about earlier. But we don't even have to look inside the fish to see that changes are happening. This is the belly view of a round goby. So the head is at the top. And they have a fin on their belly. It's called a pelvic fin. It acts like a suction disc when they want to stick to the sediment that they're sitting on. And the fish on the left is what we expect to see in a normal, happy, healthy round goby. But in a very large proportion of the round gobies that we're taking from these highly contaminated areas, we do see evidence of external damage. The fins are actually dissolving away from their bodies. And that comes when you're sitting on those highly corrosive sediments that are associated near Randall Reef. And we can also see problems arising on the insides of these fish as well, particularly focusing on males. So male fish are particularly susceptible to certain kinds of contaminants that can interfere with their hormone or endocrine systems. And in male fish, many species of fish, not just round goby, what this results in is a condition known as intersex, or the presence of both eggs and sperm in the gonads. And this is something that you typically need a microscope to see. But we can also see changes externally. So round goby happen to be a species of fish that has external genitalia. It's called a urogenital papilla. And it's really handy because we can very easily tell males from females apart without having to dissect them. So a normal female urogenital papilla looks something like this. It's kind of boxy and square and blunt. The normal urogenital papilla for male round gobies is longer and more pointed. But the urogenital papilla of males taken from our more contaminated sites is notably shorter and fatter and overall more female-like in shape. And taken together, the increased incidences of intersex and the increased incidences of urogenital papilla feminization are both signs of endocrine disruption. So we know these fish are being exposed in the field, and we know they're suffering physical consequences as a result of their exposure. If we're going to be interested now in the behavior, what should we be looking at? And we decided that the easiest behavior to start looking at was actually just round goby movement. And round gobies, again, because they can't swim up in the water column, they move by hopping around their environments. So the ABLE research group and many of our collaborators have actually brought round goby into the lab and exposed them to various contaminants in order to see what happens. For example, round goby that have been exposed to increasing concentrations of nickel show a reduced level of activity measured as the number of hops they're producing per minute. And sometimes we do see this reduction in activity over time. Sometimes we see the opposite effect, and sometimes we see no change at all. What's important to remember about all of these tests is that they are fairly short term, and they may be characteristic of that alarm phase of the stress response, because these are happening with exposures that may only be lasting a few days or a few weeks. Exposed fish in the field may be exposed for months to years at a time. The average lifespan of a goby is somewhere in the neighborhood of 2 to 3 years. If we're interested in behavioral changes that happen over months to years of contaminant exposure, there's really only one way to look at that, and that's to actually go into the field, remove fish, and test them. We decided to look at movement behavior in the context of willingness to explore, boldness. How comfortable are they in a new environment? And when gobies are placed in a new environment, they no longer simply hop around, but they exhibit these really exaggerated kind of leaping motions at the walls of the tank, and we're calling this exploratory swimming. They only do this when you first put them in a new arena. And in order to test exploration, we built them a maze. It's not a very complicated maze. It's a 2.5 meter long tank that had 5 segments. And 2.5 meters is about the size of the diameter of these fish's home ranges in the wild. We put 3 fish in at one end. Round goby are gregarious and seem to be most comfortable when tested in groups. And then we allowed them 30 minutes to see if they could solve this maze. Round goby are also somewhat inconveniently most active at night. So when we did these tests, we had to run them in the nocturnal phase of their light cycle. And in order to do this, we needed to observe them under red light so we could see what was going on. So that's what it actually looked like in practice. To catch round goby for this particular test, we went back to Hamilton Harbor to one low contamination area near LaSalle Park and 2 highly contaminated areas near Randall Reef and Windermere Arm. And what we found is that overall round goby taken from highly contaminated sites showed reduced willingness to explore. They made a fewer number of exploratory swims in the first stages of that maze. And what I have graphed here is the number of exploratory swims per minute that were being exhibited by fish from that cleaner area, that low contamination site, and fish from the 2 highly contaminated areas, Windermere Arm and Randall Reef. And you can see that their activity level is somewhat reduced. They also solved the maze somewhat less often. This is the percentage of all fish that successfully solved the maze in 30 minutes out of 100% fish. About 75% of the fish in the low contamination area solved the maze, and progressively lower percentages solved the maze from our 2 highly contaminated sites. And when they solved the maze, they were also taking a longer time out of that 30 minutes to do so. So what we know from traditional behavioral ecology research is that in many species, increased exploration in these kinds of artificial lab tests is directly related to increased movement of these same animals in the wild. For example, they may have a larger home range in the environment, or they may be more willing to disperse and disperse further in the environment. So borrowing this paradigm, we wanted to know whether this increased decreased willingness to explore in the laboratory was also reflected in the field by a decreased tendency to move in our fish from highly contaminated areas. It's not very easy to follow fish in their natural environments, and round goby are also camouflaged, and as I mentioned before, they're active mostly at night. So one of the most common ways to test or to follow movement of animals in the field is to perform what's called a mark recapture study. We would go to our sites and set a series of traps at the same locations each time along the shoreline. These are baited traps. Any fish that we caught within them were measured, And each fish was then given an individual tattoo with a substance called elastomere. These unique identifying combinations allowed us to follow that individual and know who it was for the duration of the study. And these elastomere tags are non-toxic and they last for the lifetime of the fish. We would then put these individuals back where we found them. And we would go back every week over the 2 years of the study, put traps back into the same spots, and see where these fish turned up next. So if we put a fish in down here and it next turned up there, we would know it had moved the distance of 4 traps. If we put it in here and it only turned up here, that fish has undergone a much less of a dispersal. We performed this marker capture study at the same locations in Hamilton Harbor where we pulled those fish out to give them a maze test that I just talked about a little bit earlier. And this study actually went on for 2 years. We tagged fish in multiple batches in the first year of the study, and then we followed them through to the end of the second year to see who would come back and how far they had moved. And this is kind of a 2-year time period that's defined by the lifetime of these animals. They're not going to be living longer than 2 years, especially since we're already tagging them as adults. And we can follow the movement data yielded by this 2-year study in 2 time chunks. So first we can look at movement that was produced over the first year of the study. We can look at the distances that fish moved from their first tagged location to their last known location, and the number of fish that, the total number of fish that did return to our traps is graphed here along the y-axis. So what we can see is that over the first year of the study, most of our fish were actually not moving very much at all. They moved only 0 to 3 meters from the location where we last saw them. A few more moved 6 to 9 meters. Even fewer moved 12 to 15. I think maybe we only had 2 individuals who moved 18 to 21 meters from where we last saw them. But when we look at differences in sites, we actually didn't see any difference at all in how fish were moving in these low contaminated areas and these highly contaminated areas. We went back in the second year of the study to see who came back. And we can now track the movements of these individuals from where we first saw them in year 1 to where we last saw them in year 2. And again, we can graph the data as the amount of distance these fish have moved over the whole time that we've known them versus the amount of fish that we successfully returned back to our traps at all. And what we can see again is over 2 years, these fish are moving a little bit more than what they were moving in the first year of the study, but we're still not seeing any differences in how these fish are moving in areas of low contamination and high contamination. So this is not what we expected. So what's going on? We know that round goby living in these highly contaminated, very toxic areas of Hamilton Harbor do show a lot of physical signs or biomarkers of contaminant exposure. We also know that when we take them out of their home and bring them into the lab and give them an artificial test like a maze, that we do see behavioral differences. And these differences are repeatable. But we also know that round goby living in these contaminated areas are very abundant. Their overall numbers don't seem to be impacted too much by the contaminants in these areas. So what we suspect might be happening is that there has been sufficient time for behavioral compensation of these fish. They're able to make any adjustments for long-term impairments in movement behavior because they're now well within the resistance phase of that time of the stress response. They've been exposed for months to years, and they've been able to adapt to that. The general concern is that these highly successful invasive fish are also highly pollution tolerant. They're able to live in some of the dirtiest spots in Canada, and they seem to be thriving there. And this leads to an additional worry. Since the round goby has become so well established in the Great Lakes, it's now a prey item for a lot of the predators. And if goby are able to live in contaminated sites, they're prey items. So they may be drawing predators to those particular areas and facilitating mobilization of these pollutants upwards through food webs. So I'd like to just end here with a little bit of a prospectus to how the field of behavioral ecotoxicology is going to be moving forward. Overall, toxicology is very, very good at understanding acute responses or short-term responses to toxicant exposure. We've got a pretty good handle on also the kinds of behavioral changes that happen during these short-term tests. And in this particular area of research, what scientists are interested in doing is getting a better understanding of what the mechanisms are that are actually causing these behavioral changes. Knowing the sequence of steps that happens from an initial chemical exposure through to a change in behavior is going to be really useful in continuing to use behavior as a kind of sublethal biomarker. And there's also been a lot of recent interest in how to make it easier to observe and record animal behavior when you're running a lot of tests repeatedly over and over and over again. And there's been a tremendous push towards automating animal observations in the laboratory using software or fish tracking devices to make it easier to collect these kinds of data. When it comes to understanding the ecological relevance of the sublethal changes that we're seeing in contaminant exposures, there's actually a lot less data available to us. We simply don't have enough studies that are working to address the long-term consequences for animals that are being exposed to contaminants. Are they showing evidence of resistance or developing tolerance? Are they showing signs of impending ecological death? Or we don't yet have enough information or enough studies that are attempting to address this. And scientists in this field generally feel overall that we need a research paradigm that emphasizes longer laboratory tests and comparisons with field data where possible. And part of the issue with this is that ecotoxicology is still a young and developing field. There isn't always the research incentive there to run these types of tests. But it's going to be increasingly important if we want to be able to recognize developing problems in the environment while there's still time to fix them. I'd just like to take this moment to thank everyone in the ABLE research group and elsewhere in McMaster, as well as our collaborators at the Canada Centre for Inland Waters, where I currently work, and the Royal Botanical Gardens. So many, many people have contributed to this research and allowed it to actually happen. Thank you very much for your attention. I'm happy to take questions.